Just a few, okay, yeah. So just a few quick notes. Today we won't have the quiz, but feel free to post your questions in the chat box, which we'll ask Ian after his talk. So Ian is head of science at National Physical Labs. National Physical Labs is probably one of the oldest physics institutes in the UK and does everything with meteorology. If you remember what a kg is, what a meter is. So I just joined NPL, and he's a senior NPL fellow and also a visiting professor at Nottingham. So he's also the founding director of UK National Center for Excellence in Mass Spectrometry. So Ian develops super resolution label-free metabolic imaging, about which we will hear in the beginning. And then he's also leading the latest developments at Rosalind Franklin Institute. So Ian, looking forward to your talk, and yeah, quite excited.
Okay. Great. Well, thank you very much to you all for the invitation to give a talk. And what I'd like to talk to you today about is kind of an introduction to biological imaging using mass spectrometry. And then I'm going to tell you about some of my own research where I developed this instrument called the OrbiSims, which is for my research goal is to try and get to super resolution metabolic imaging. We're a bit of a way off that yet, but we're getting closer. And then in the latter part of the talk, I'm going to tell you about the Rosalind Franklin Institute and what they're doing there to build a sort of a really next generation instrument as a national capability. So I'm part of a Cancer Research UK Grand Challenge team, Rosetta. So you see I have many collaborators, and I think many of them from institutes of people on the talk here. And it's a fantastic opportunity really to develop these techniques that are biologically relevant and useful. So my own research is very much around supporting the pharmaceutical industry and particularly this challenge of drug attrition. So most drugs will fail through the pipeline, maybe only 5% or so will eventually get through. So there's a huge kind of effort to try and reduce these late stage failures, because if a drug fails at late stage, remember the process is 10 to 15 years long, the costs by then are enormous. And there's this kind of guiding philosophy for that is the so-called Pfizer 4 pillars. So these are these 4 pillars. Now, if we think of them, most medicines are delivered orally. So if we imagine this medicine here, and we're going to take orally, and let's say we're going to try and target this piece in the intestine here. So here's the epithelial wall, and let's say, for example, that these cells here, these epithelial cells have gone cancerous. So you can see some of these are changed. We want this drug, which will be systemic, to eventually reach the biological target, which is usually some protein, let's say in this cell. So the first 2 pillars are, does the drug get to the target? So just do we actually get enough of the drug to the right place? The next pillar is then, does that drug actually bind or modulate that particular protein? The third pillar is, do we get the right functional pharmacology? So if we understand the disease mechanism well, are we then getting the right metabolic changes at the tissue level? And the fourth pillar is that the studies need to be done in tissue, in phenotypically relevant tissue. So that's a big change in pharma these days, in increasingly trying to do drug development, drug screening on human tissue where possible.
Someone's for James, Nick.
Hello? Yeah, you can hear okay?
On that table there. Cool. Cheers. Okay.
So mass spectrometry imaging is really a fantastic tool for this, because as I'll show you in a little while, we can get molecular level imaging, and we can use finely focused probes to be able to get that resolution down to the single cell scale. So we can see if drug has got to the cell. We can't actually really tell with mass spectrometry imaging if a drug is bound with the protein, so we really have to rely on other techniques for that. The third pillar is we can follow these metabolic changes as I'll show you. And the fourth pillar being phenotypically relevant. Well, that's perfect for us because we can do imaging on human tissue, no problem. We can't do it in vivo, that would be very nice, but we can do it on human tissue. So where are we in the grand scale of sort of omics? I mean, of course, there's been a huge revolution in the omics in terms of looking at the single cell scale. And, you know, why is that? Well, if you look at this kind of cartoon of tissue here, we see all these different cell types, and then even cells which are of notionally the same type, it's understood now actually are very different. There's a high heterogeneity in these cells, and this can be understood at the genomic level. So you can now do, using next generation sequencing, you can get genomic information down at the single cell scale. In transcriptomics, phenomenal developments in techniques like FISH-seq and MERFISH allow getting kind of pretty deep transcriptome coverage for the single cell scale up to something like 10,000 cells. And these techniques have done so well because they can make great use of these kind of barcoding techniques, PCR, of course, amplification strategies, and that gives them the sensitivity. When we get to techniques like proteomics, well, then we need to use methods like mass spectrometry to be able to identify what that protein is. And at the moment, there just isn't the sensitivity to get that down to the single cell scale. If you have an antibody, of course, then you can use that in combination with a reporter, either an optical reporter, so you can then use super resolution techniques, or you could use a metal reporter and use laser ablation, ICP-MS, CyTOF type instruments, and then follow the proteins. So genomics, transcriptomics, proteomics, in metabolomics, well, in metabolomics really is difficult because we can't use any amplification strategies because the molecules are just spontaneously generated. We need to use a technique which has got very high chemical specificity because a molecule may only vary by subtle change in a bond here or there. And consequently, techniques like mass spectrometry are really the main ways you would look at metabolite information. But how do we get that down to spatial resolution of the single cell and higher resolution than that? And that's a big focus of my own research. So how does mass spectrometry imaging work? Well, we normally have a tissue slice. So here, for example, on a sample holder. And then we use some kind of ionization probe. Now, these probes can be laser probes, they can be chemical probes like an electrospray probe, or they can be an ion beam probe, which are focused at the sample. And then we raster across that sample in some kind of pattern, so the computer knows where the probe was. And then from each of those points, we desorb material. Now, the big challenge in our field is that very little of this material that's desorbed is ionized. We have to have it ionized to be able to detect it in a mass spectrometer. I'm not going to go into the details of that in this talk, but that is a challenge for us. But from this, we get this rich spectrum of material that's come from the surface. We record that in terms of the mass to charge and the intensity. And because the computer knows the position, from each time we fire something and we collect it, we can then reconstruct in the computer images for particular masses. And then depending on the type of probe we use and the type of mass spectrometer we use, we get different levels of spatial information and chemical information. At the National Centre of Mass Spec Imaging that we have at Imperial, we have a very wide range of mass spec imaging techniques. And I won't be able to talk about them in detail, but hopefully give you a bit of an overview. Techniques like MOLDI, matrix-assisted laser desorption, that uses a photon probe, a laser beam. Desorption electrospray ionization uses, over here, uses an electrospray probe. I'm going to talk quite in detail today about SIMS, secondary ion mass spectrometry, which uses an ion beam probe. We can focus these down to very high spatial resolution. And then there are techniques like in situ surgical based methods called REAMS, rapid evaporative ionization mass spectrometry. So you can then, for example, while you're cutting through a tumor, you can get an online mass spec readout. So there's a really broad range of techniques. Okay, this plot is a little bit detailed, but I hope I can just kind of guide you through it because there's always a sort of a trade-off. Now, on this axis here, I've got the amount of fragmentation that that we get in a different type of ion beam combination. So this here is using a technique called nanosims. So here we use a monatomic ion beam, which we can focus down to 50 nanometers or so. But the downside of that is if you look in the mass spectrum, the black signal you see here is like the molecular signal. You see we get no molecular signal, we only get fragments. So we're kind of, in biological studies, we normally use some kind of isotopic labeling strategy. But with that labeling strategy, we can get down to fantastic resolutions of 50 nanometers. At the other end of this scale, so this axis here is the spatial resolution. At the other end of this scale, we can use very gentle techniques like DESI, desorption electrospray, which uses an electrospray probe. And like electrospray is a gentle ionization mechanism, and you can, in fact, look at intact proteins. So you can see the molecular signal without any fragmentation, but the trade-off is now we only have hundreds of microns of spatial resolution. And in between that, we have varieties of different ion beam combinations. And what we're all trying to do is push as far as we can into this corner here, where we have hundreds of nanometers of spatial resolution, but very little fragmentation. Because if we're looking for metabolites, for instance, we don't want to create fragmentation from the ion beam, because that could be confounding factor in terms of trying to identify a particular metabolite. Now, in terms of what gives us the best combination here, it's actually using ion beams which are focused large clusters. So here in the nanosims, we use a single cesium ion in the beam. For biological imaging, we now use thousands of argon atoms which coalesce into one projectile. And technical limitations mean with this, we can get down to about 1 micron in spatial resolution. And that's currently kind of the state of the art in terms of getting, sorry, just trying to get the cursor to go, the laser light to go, in terms of getting a high spatial resolution, but not getting too much fragmentation. So this is kind of what I'm going to talk about in the first part of this talk. So what are the current state of the art instruments in secondary ion mass spectrometry for organics? Well, there's a variety of different vendors who will sell you an instrument, and they're all very powerful. They all have mass spec kind of capabilities these days, but they have detectors which are based on a time of flight mass spectrometer, and that is, turns out not very good for identifying molecules, and I'll show you why. So this is a portion of a mass spectrum of, you would get, if you took some brain homogenate, and we were able to analyze this with one of our imaging instruments. This is the kind of mass spectrum we would get. So this is from the range 154.05 m/z to 154.13. So it's quite zoomed in, as you can see. And even though we're zoomed in a lot, you can see there are many different molecules. Now this is recorded with the type of mass spectrometry you use in a metabolomics laboratory, which you'll have in your institutes. But if we overlay that with the mass resolution that we would get from one of these time of flight mass spectrometers, you can see the peak is very broad and would integrate many different biological peaks. So for instance, if we were interested in dopamine, which is a low-level neurotransmitter, the intensity of the peak is low, we would confound that with these other peaks here. It would be no use. With these kind of modern spectrometers that I just showed in the previous slide, we can actually get that around to about 10,000 mass resolving power, but still is way off being able to give unique identification of these metabolites. But it's actually even worse than that, because not only are the peaks broad, the accuracy of those peak positions is not very accurate. So the peak could be kind of anywhere within something like 30 to 50 parts per million window. So it really means with those type of instruments, they're very good if you know what you're looking for already, and you can have confidence in identifying it. But if you don't really know what it is, or you don't have confidence in identifying it, and for example, if you're working in the pharmaceutical industry and you're making £1 million decisions, you've got to be very clear and very sure that what you say you think it is. So this has been a problem for the field for some time now. There are other types of mass spectrometer that you can use, and this is an example here of the so-called Fourier transform mass spectrometer from Alan Marshall in the US. Here you can see the peaks are super sharp, and in comparison is the same spectrum taken with a time of flight mass spectrometer, where you can see everything is confounded. So the trouble with these time of flight SIMS instruments is that sensitivity is too low. There's too much fragmentation. The mass resolution and mass accuracy are too poor for identification. So if you're lucky enough to detect a signal, you'll have no clue what it is, and it's probably a fragment of something else anyway. So that's not great. So some years ago, I sort of tried to find a way around this problem, and it is a tricky thing to do because, again, this plot here helps us understand why. So on this axis, I've got the time it takes to take a spectrum using different types of mass spectrometer design. So there are lots of different types of doing a mass spectrometer. And on this axis here, I've got the mass accuracy. So this is, remember, this is the parameter that we want to tighten up so we can be more confident. And unfortunately, if you want really good mass accuracy in the green zone here, the types of spectrometer you use for that are these so-called Fourier transform instruments. But they're really slow. They take like a second or so to take one mass spectrum. Well, if you're imaging and you've got 128 by 128 pixels, you can see on this axis here that you'd be at 16,000 seconds to take one image. Well, you know, a whole PhD could be taken up by just taking a few samples. So that's really not going to be very practical. At the other end of this thing here, this plot here, we have these time of flight analyzers, which are what we use. And the reason we use them is because they're fast. So we could take 128 by 128 pixels in 16 seconds. So they operate very quickly, but say the mass accuracy is not very good. It's about 30 ppm. And the problem is there isn't like one instrument which would allow you to get down to high mass resolving powers, high mass accuracies, but also do it fast. So this is where I came up with the idea of making a hybrid instrument. So what we did is I took one of these Fourier transform instruments, so-called Orbitrap, which is very often used in metabolite ID labs or protein ID labs in biological mass spec, which has kind of is relatively slow, so we can run at about 10 hertz, but it does give us mass accuracies of around part per million. So that allows us to get down to this kind of point. And if we combine that with a time of flight analyzer, we can then use the time of flight analyzer to image things fast. And the idea then is that we then have an instrument using both analyzers that kind of support the mutual benefits and disadvantages of the analyzers. And it's a very similar plot for this other parameter of mass resolving power. It's a little bit more complicated because instead of points, you have lines, but I won't get into the detail of that now. But again, if we use just the time of flight analyzer alone, then we have mass resolving powers of really only around 10,000. If we combine that with the combination of an Orbitrap mass spectrometer, then we can get mass resolving powers of several hundreds of thousands, even 500,000. So that was the concept of building this OrbiSIMS, and I'll just give a little kind of quick overview of what we did. So this was published in Nature Methods already in 2017, so it's been developed a few years now, and there are actually 10 of these OrbiSIMS instruments around the world now in different labs. It's being commercialized. But this is ours back in 2016 as we launched the first one. So it was a joint project with GlaxoSmithKline because they were very, very keen to be able to do, as I showed earlier, see where the drug goes. That pillar 1 of the Pfizer and pillar 3 is the drug at the target, and is it having the right pharmacological effect? Driven a lot by Sir Colin Dollery was really, so he was a senior advisor at GSK, and Sir Keith Peters, who was at GSK and then has been a strong figure in the development of the crick. These really push me actually in terms of, you know, the current instruments are not good enough, you need to do better. And it kind of came at a time when I had this good idea, and well, we built this instrument. So The instrument is kind of complicated, and I don't want to go into the detail of that, but the advantage of these two analyzers is that we can operate in many different modes. So we can operate in a depth profiling mode. We can acquire chemical information as we go deep into the material. We can just take surface spectra, we can do 2D imaging, and we can do 3D imaging. And I'll show you some examples of that. So this is a very nice example, really, of what you can do, because let's just say, if we take a piece of, this is a mouse brain here, and a typical H&E pathology image, what we can do is put the same sample into our instrument. We can do a fast image using this so-called time of flight analyzer. So remember that this operates fast, but the biological information from it is not very good. But we can use that then to identify a region that we're very interested in. We can then switch to this Orbitrap analyzer, which is now slow, but gives us this wonderful high resolution mass spec imaging. And now we can see single cell level detail, but every pixel in that is a mass spectrum. And we can identify 70 different lipids, all with different metabolites, serotonin, dopamine, with around micron spatial resolution, so subcellular resolution. And then we can overlay this with optical methods. Another very nice feature with this instrument is that we can trap the ions and we can fragment them and break them apart. So we can look at the structure of these molecules to give more confidence in terms of the identification. And we can image at high resolution. So this is, these are rat alveoli macrophages, these NR8383 cells. And we've incubated them with a drug amiodarone. And what's very nice about this is that we can image these cells in 3D. We can detect the drug within the cells. So the drug is in green, a marker for the nucleus is in red, and then blue is a marker for the cell membrane. What I'll show in the next slide is we can then, this is the amiodarone incubation concentration going up to 10 micrograms per milliliter. And then these are individual cell measurements of the amount of phosphocholine that's been upregulated, this 34-1 lipid. And one of the things that really struck us when we first did these measurements was how scattered they were. And as a physicist, I thought, oh, there's something really bad with the machine, there's something wrong. So we ran, obviously, reference samples. We found the machine was good. It was repeatable, very good repeatability. Then what we realized is that this was actually biological variability amongst the cells. And the nice thing with the technique is not only do we measure for each cell changes in the metabolites, but we can also measure for each cell the amount of amiodarone that's been taken up. And again, you can see there's this huge variation. Some cells hardly take up any amiodarone, some cells take up lots of amiodarone. And the nice thing is you can then plot this for cell by cell data. So on this axis, we have the amount of the signal relating to the amount of amiodarone in the cell, the amiodarone intensity, and then the phosphocholine intensity. And you can see these all now fall on one kind of roughly straight line. So there's this high heterogeneity within the cell population, although they're all following the same sort of biological mechanism. And actually, we use this cell and drug system because it's well known to call this so-called phospholipidosis, that the drug goes into the lysosomes and these form these multi-layer lamella. And here we're able then to see particular lipid markers or biomarkers which would be indicative of this effect. So it's very nice to be able to do that at single cell scale. Now, I'm sure for microscopists, and I see there are people from the LMB here and from Cambridge will know very well that cryogenic techniques are critical for being able to look at things in biological detail. And everything I showed you so far has been freeze-dried because that's what we could do at the time. But we realized that, that's for looking at biological systems, that's not really very good. We need to be able to look at things in their native state. So we kind of shamelessly just took the technologies that the cryo-EM community have been developing and then re-engineered those into our instrument. It turns out we had to do some things differently because we have to ionize the molecules that that come from the surface to be detected, and some of the cryoprotectants used in high pressure freezing, in fact all of the cryoprotectants used in high pressure freezing killed our signal, so we lost the ionization, but we did manage to find one cryoprotectant that worked. So we tested this out using cryo-orbisims and of a biofilm. This is a mature Pseudomonas biofilm. These are really complex structures with the bacteria that are protected in this polysaccharide scaffold. And we're very interested in the sort of quorum sensing signals, and can we sort of do label-free imaging of these? So this was actually quite a job, and my postdoc Junting Zhang and colleagues really did a fantastic piece of work to develop up the system to be able to do this. And we have a very nice sort of cryo workflow, which I think is comfortable for those working in biological sciences, because if you're used to using these Leica systems, you can then take the sample directly using these Leica sample transfer shuttles into the orbisims and do cryoanalysis. And here's an example here of a 3D image now of one of these biofilms. And the nice thing with this instrument is we can get 3D molecular information. So here we're following a potassium marker, which is one that will show us where the bacteria are. But we can also look, for example, where in the next slide, we can follow quorum sensing molecules and look at those layer by layer to generate a three-dimensional structure. I collaborate a lot with the Francis Crick Institute, particularly Alex Gould's group, and we have a joint student, Claire, and Claire's been doing fantastic work on developing the cryo-orbisims for looking at Drosophila and also other biological systems where you want to look at molecules that otherwise will be lost because they're semi-volatile. And this is Claire's paper that was recently published in Angewandte, where by using genetic knockdowns, he was able to knock out a particular enzyme, which then altered the amount of tricosane and tricosine at the surface, and we can follow these metabolic changes and see exactly what she was expecting to happen from the genetic study. We can see that translating into metabolic changes at the fly surface, which is really very cool. We can also use this cryo-orbisims, for example, for looking at fingerprints, getting metabolite information on the fingerprints, and identify all sorts of different molecules that normally would be lost in the vacuum system. And also in pine needles, for instance, Claire had a great idea to try this technique and look at different plants and identify then molecules and see them really for the first time, these semi-volatile molecules in along with the sort of the plant structure around these stomata. So that kind of shows you the sort of the work that I've been doing with the cryo-orbisims. And then the next thing is, well, where next? And this is kind of leads on to the next part of this talk and the work at the Rosalind Franklin Institute. So I'm sure many people are aware of the Rosalind Franklin Institute. It's a new national institute that's currently being built, and I think probably not so long before it will open. And it has 5 themes within it, which are looking at 3D structure, the time domain and chemistry, so structural biology, time-resolved imaging, next-generation chemistry, artificial intelligence and informatics, and then there's a mass spectrometry imaging theme. And the Rosalind Franklin Institute has a whole range of different routes for collaboration, so people could bring samples to the different technologies that have been developed in the Franklins for proof of concept experiments, joint grant applications. There's a potential for placements, collaborative students, and industrial collaborations using one of the many technologies that have been developed. And it's funded by the UKRI, EPSRC. And these are all the sort of member institutes that are part of that. And MPL, Josephine Bunch is co-leading the mass spectrometry imaging theme with Zolatan Takats from Imperial. And the aims of this biological mass spectrometry theme is to create a platform which enables the identification of signals detected by mass spectrometry imaging and in situ methods for improved multimodal imaging, improved sensitivity, and comprehensive suite of ion structure elucidation methods. And also they're very keen to integrate this with other imaging themes of the RFI, for example, electron microscopy and cryo-electron tomography, correlated imaging and photoacoustic imaging. And they have a kind of a very grand challenge, which is to be able to do molecular level information at tens of nanometer scale. So I think a fantastic challenge. And what they've done is brilliant in terms of bringing together the whole community. So they've done a lot of consultation with the universities, with the pharmaceutical industry, with biological scientists to sort of design what would be the kind of the dream instrument going forward. And from that, you know, they've then got a really powerful design based around different sample introduction methodologies, cryo sample preparation, different probes. So, you know, each probe is important for different types of things you want to analyze. Electrospray, for example, for looking at proteins and moldy, for looking at large molecules, SIMS for looking at high resolution, but with more fragmentation. And then a really powerful type of set of mass spectrometries that can be used, including looking at the shape of this using ion mobility, and also looking at Oxford at fundamentally new methods of doing detection using pixel imaging detectors. So as I said earlier, this new instrument will have different probes. SIMS, as I've talked about extensively already earlier from relating to my own research, but it's very much moldy and desorption electrospray ionization probes will be possible on this instrument in this kind of combined type sample analysis platform. And I think a particularly powerful feature of this, because with the Orbisims, we know, even with all the power in the Orbisims, we can't always separate things out, because if a molecule can have the same mass, but can have structural isomers, so can have 3 different shapes all with the same mass, we can't separate them. But using the ion mobility, which would be in this instrument, you can see in this example here, that these 3 molecules have exactly the same mass, but they've got just different structural arrangements. And their shape, they'll have a different drag in a field, and that can be used to separate these out. And then once you've separated them out according to their shape, fire them into the mass spectrometer to get the mass spectrum. So overall, the kind of design concept of this instrument is like this. We can use, for example, this SIMS platform. So this is Ionoptica. They've been leading a lot of the development of the instrumentation around this, which gives you the high resolution imaging. We combine that in with sources, for example, like electrospray and moldy. These can then go into this very sophisticated mass spectrometer based on a Fourier transform instrument. So this is being led by Bruker in terms of the instrument development collaboration. So these instruments go up to very high mass resolving powers and mass accuracies, which I try and show on this plot again. So this was the Orbisims. Here we're looking at the mass resolving power. So you can see by combining an Orbitrap with a time of flight SIMS instrument, we were able to get to some hundreds of thousands of mass resolving power. In the Franklin instrument, what they're doing is they're combining this now with a Fourier transform instrument, which will give you orders of magnitude improvement in the resolving power, taking you up to tens of millions nearly of mass resolving power, and also improving the mass accuracy. So this instrument will be an incredibly powerful machine. So that brings me on to my conclusions. So mass spectrometry imaging is a really powerful technique for label-free molecular imaging. The Orbisims allows imaging with 1 micron mass accuracy and a mass resolving power of 240,000. And we have this MSMS to give fragmentation, so we can look at molecular structure. And the cryo-Orbisims allows imaging of molecules in their native state using high pressure freezing or plunge freezing protocols. And I really think this is important, because if we want to now do correlative imaging with, for example, light sheet microscopy, that's something that we're very interested in being able to do, for example, of an organoid. If our image is of a collapsed organoid and theirs is a nice hydrated system, it's going to be very difficult. Where now, hopefully, we can get 3D imaging with the cryo-orbisms of the metabolites and even different drug molecules where they've gone within the organoid, and then overlay that with really high resolution structural information and labeled information from light sheet microscopy. And the Rosalind Franklin Institute is developing a next generation mass spectrometry imaging, and this will give orders of magnitude improvements in the mass accuracy. That'll move that up to something like 200 parts per billion, mass resolving power of 10 million, ion mobility for looking at the molecular shape, and also multiple source probes, laser, electrospray, ion beam for the analysis of intact proteins to small molecules. So with that, I'd like to thank you very much for your attention, and open to questions.
Thanks, Ian, for the great talk. Just impressed by how much you have developed at NPL. We have a few questions, and what we usually do is I also invite participants to ask the questions themselves. I will start with the first question, and then we go to Stephanie, and then Pritish, if you want to ask, and yeah, as more questions come in. So you mentioned about the correlative imaging, and I wanted to know your thoughts about, labeling procedures. Let's say if you wanted to do 3D orbisims with a light sheet, like what kind of common labels you can think of that you can use as a proof of principle, let's say.
Yeah, that's a good question. So we've done orbisims analysis, and then after that, we've taken the same tissue sample and labeled that with things like DAPI and other stains that you would often use in immunohistochemistry, and that's worked quite successfully. We haven't yet done the other way around yet, and then had the labels that you would use in light sheet microscopy and then done the mass spectrometry imaging. So that remains to be tested. We do worry often that labeling strategies might kill our ion signal. So there may be subtleties in that, and that needs to be tested and checked out, because if you do something which becomes a kind of, it's all about competitive ionization. So if something wants to ionize more than one of the analytes you want to detect, then that could lower or kill your signal. So that is something that needs to be checked out systematically, really, which is kind of work that we're currently doing with colleagues at NPL, with Mike.
So just touching on the same point. So let's say like you would like to image the same structure, right, across different, let's say, orbisims and then light sheet microscopy. So let's say like DAPI, right, like you mentioned. So what I understood, you add DAPI later on, but you would like to image the same nuclear structure and then somehow translate it to the same, let's say, spatial map of DAPI that we get from light microscopy, right? So
yeah, exactly. So I mean, the goal would be to be able to take an organoid, for instance, or piece of biological tissue that had been imaged by light sheet microscopy using whatever labels you want to show the structure. And then we would, on the same sample, be able to do a 3D molecular image to then, for example, look at metabolites. So we could paint over your image where different metabolites are, where different drug molecules are, albeit at lower resolution, but with that registration of those images. So that would be the dream. We may.
My question would be, let's say like, this is the last of my questioning. So let's say like you want to study how a drug gets delivered in a lung tissue, right? And then you use your method, and then you also want to visualize exactly the same drug. Can we see how it locates in a given tissue, in which part? I think, instead of, adding different dimensions to the same image, let's say DAPI and metabolites, you just want to study one specific gene delivery, drug delivery, something like that.
Yeah, sorry, I'm not quite sure I understand the question.
Okay, yeah. I think I will ask you later on more.
Okay, sure.
And she has a question on Raman. So, Stephanie.
No, it's just kind of wondering, I mean, listening to it, obviously, I'm like amazed how far it's correlating with existing techniques. And I mean, I was just cheekily glad you're not reaching the resolution we have in light microscopy yet, because I would be jobless. So I think I got I think it was 500 nanometer, which was mentioned, you kind of achieve with some of the imaging. And so I was just curious, I mean, how big are the systems? I mean, if I wanted to buy a system and establish it in my lab, is it as like a cryo-EM, or is it like a confocal potentially in terms of implementation? And then how do we, you know, how in future, will we just buy your systems and then we kind of forget about light microscopy?
It's a good question. I think to take that last point first is
it's a bit of
forgetting about light microscopy, because I didn't get time to go into the detail of the mechanisms, but because we're a desorption technique, every time we improve the spatial resolution by a factor of 10, there's 100 times less material for us to analyze. So there's 100 times less signal. So we have a kind of problem as we crank up the resolution, we're fighting sensitivity all the time. So like my real goal, research goal, is to get down to like 100 nanometers. Currently we're about a micron resolution for label free. My goal will be to get down to 100 nanometers, but that means I need to get 100 times increase in sensitivity. And that's actually difficult. Now, funnily enough, going to cryogenic samples does give us a boost in sensitivity. The water acts as a kind of a matrix, protonating, improving our ionization. But we're really having to look at something called post-ionization lasers to convert the neutrals. Most of the stuff we get off the sample is neutral. We need to try and convert that to ions. So that's a big focus of kind of research that we're doing. Now, in terms of the kind of setting up the instruments, I mean, things like the Orbisims are pretty expensive because, I mean, it's got 2 mass analyzers, a lot of ion beam imaging. So you're into sort of the €2 million kind of mark for setting up an instrument like that.
You also need the whole infrastructure for like cooling and gases and all of that as well, I guess, no?
Yeah, so yeah, you would, if you're using the cryo side, you would need liquid nitrogen and things like that. Having said that, you know, it's much a standalone piece of kit, like setting up an EM or something. You know, it's an expensive piece of kit, but it's very versatile and does many different things. At the other end of the scale, there are techniques like the DESI technique, which is amazing. Biological information you get from techniques like DESI is really excellent. But the compromise is the spatial resolution is much lower. So it can be, for example, another way of doing it is you buy one of the cheaper type instruments like DESI, which maybe you could get for, I don't know, but maybe 300K or something, quite a nice setup. And then you just go to a national resource when you need higher resolution. So, for example, that's kind of one of the ideas with our National Centre of Mass Spec Imaging. Not everybody can be buying these expensive instruments, but by having them in one location with the experts to run them, because actually that's also part of the complexity. I mean, they're not trivial instruments to use, then people can come and, you know, get access to those machines when you need that kind of next level of resolution or sensitivity.
But the imaging, your imaging is always the last, because there's then the samples kind of disappear.
Yes,
so
we're
it's
always
kind of,
desorbing, yeah.
But yeah.
I was
going
to say, with the other end of the resolution scale is this so-called nanosims, where we kind of need to use a label, so we use an isotopic label, but then we can go down to, I think 35 nanometers is the best we can get, which is is really nice. It's very complementary to EM. The downside is you do need to have some kind of labeling strategy.
I mean, for the drug imaging and in tissues, you don't probably need the resolution. It's not the limit, actually, I guess. I mean, for many applications or questions.
Yeah, I think, well,
it has to be.
Very often we need to know whether a drug's gone to a particular organelle. You know, as well as the knowing off-target effects. So if a drug ends up in the mitochondria, then that can often be you know, a very bad sign. So we need to be able to identify well.
I was also wondering about the cars imaging, because we had that talk just before on cars imaging in terms of, you know, competitiveness or kind of correlation on, I don't know if there's any, it's a bit of a, I haven't thought it through as a question, but do you have
No,
some...
I think it's really good. So we have SRS, which is similar at MPL, and that's a really active of our work at the moment, is to try and do correlative imaging of the two techniques, and each one shines a different light on the sample. You know, the SRS, those techniques are very good for sort of looking at where those different lipids are at high resolution, and then with the mass spectrometry, we can identify what those lipids are with nice specificity. So I think always these techniques bring something else to the table that on their own they miss.
Because I guess historically NMPL was kind of, I mean, lasers are abundant and all kinds of lasers are kind of probably available for exploration, and that's the key for quite a few of those techniques.
Yeah, very much. And I think actually that's a very nice thing about the Rosalind Franklin Institute as well, for developing this next generation instrument, because you have the laser pool there, a lot of laser expertise. And I think that's something that will be important, particularly for this post-ionization thing, where we need to convert more of these neutrals to ions.
It's nice, actually. The Rosalind Franklin worked with an apparatus which was made by one of the LMB alumni I used to have lunch with, Uli Arndt. And it was such a lovely friend. Yeah, he died now 10 years ago, but I mean, we often had lunch with him. He was telling us about how he created the system which Rosalind Franklin used.
Oh, amazing.
It's really lovely.
Amazing piece of history.
Yeah. So there's some other questions, actually. Sorry, I could ask 10 more, but it's fine. I mean, other people want to ask too. So
how we will do it is like Pritesh and Sherry Huang and Catherine and others. Can you? We will ask you to unmute yourself. So in the order of the questions you have posted, and then you can have a go. So Pritesh, do you want to ask your question yourself, or shall I ask for you? I think all of you can unmute and also show your lovely faces if you want. You should be able to do that. Kate tells me, so that should be possible. Anyways, Pritesh, so the next one is I think Cherry Huang. So I will ask questions from Pritesh. He has two questions. The first one is this imaging technology gives more details of cells or tissues than super resolution microscopy? And his second question is, can live cell imaging possible by using this technique?
Live cell imaging, not with the orbisims for sure, because, well, I mean, you could high pressure freeze it and then maybe or not the cell could be recovered, I don't know, but certainly you couldn't look at something that was living at the time, so to speak. Now, that's with the orbisims. With this, one of the other techniques I call reams, then if you can do that, I mean, it is destructive, of course, as a technique, because we have to get molecules off, but you could do that on something which is alive.
So the next question is from Cherry Huang. Do you want to unmute and ask question yourself? But this was more along, Ian, what I was trying to ask, how you reconfirm the information given by orbisims, if it was correct, and can we have a good ground truth by typical some biochemical methods?
Okay, yeah, no, that's a really good question. So we can always do a tissue homogeneous sample and put that through LCMS, which doesn't give you the spatial distribution, but does give quantitative information on what molecules are there. And that's a relatively standard thing for us to do in biological studies. We'll have usually the pharma companies or our biological collaborators will run standard LCMS type experiments. So that gives you some good assurance that we're looking at the right things. And in terms of the structure, then we can tie the image in with other optical imaging techniques or scanning electron microscopy imaging of the sample. So for example, the work we've done on the amyodrone I showed you with the Orbisims, that's kind of one part of the story, but we've also looked at the same sample systems using nanosims and electron microscopy. And with the nanosims, we could actually see this particular drug, amyodrone, has iodine in it. So we could actually track the amyodrone from the iodine signal going into the lysosomes. And then with the electron microscopy, we could then see these onion rings forming around the lysosomes. So again, it's kind of a combination of techniques which gives you that information. The Orbisims bring in the molecular information about the upregulation of those phosphocholine lipids, the electron microscopy, and the nanosims giving that kind of localization and structural information.
The next question is from Ronel Roth, and the question is it possible to do MSI on 10 sections of the tissues that have been HFP and FF in resin TEM20? A lot of acronyms, so I hope you got
Yeah, so usually for the for Orbisims, the resin embedding kills our sample signal because you've got cross-linking of proteins and materials like that, so that kills our signal. But for Josephine and Zolatan have been developing some very cool techniques as part of the Cancer Research UK grant, which allows them to use laser-based methods to still be able to image lots of rich molecular information from such samples. So in the case of Orbisims, the answer is probably no, but for other techniques, especially the ones developed by Jose and Zolatan, that it's still possible.
The next question is from Catherine. Catherine, you want to ask the question yourself?
Hi, I'm hoping you can see me.
Yes, I can, yes.
Myself, so right. So I have actually two questions. The first is that you mentioned in your talk about having to find only one particular cryoprotectant that really.
Oh, yes.
Could you explain a little bit more about that, and then I'll follow on with my second question?
Sure. Yeah. So we tried all of the sort of normal cryoprotectants that you would use, and whilst they obviously protected the sample really well, they killed the signal. Now, as I was saying, when we desorb something from the sample, about 1 in 10 to the 4 or 1 in 10 to the 5 of them are ionized. So not very many of them. And unfortunately, these cryoprotectants were preferring to be ionized than our molecules. And we have a sample preparation protocol that we use called with some ammonium formate. Ammonium formate gets rid of salts. Salts kill our signal. And it serendipitously seems to work quite well as a cryoprotectant. So we switched over to using ammonium formate as the cryoprotectant, and then everything worked fine. So it was one of those moments when we were like really a bit in despair because everything we tried seemed to fail. And then we tried this one. We actually got a lot of help from NIBSC, Kirsty McGowan Gibson, and they have a lot of EM experience, and they confirmed for us that this ammonium formate was working well as a cryoprotectant. So it was, I think we kind of got lucky, but it was after an awful lot of frustration and tears.
Empathy from a crystallographer we've struggled through years with trying to find good cryoprotectants and understand some of those frustrations. My second question really has to understand a little bit more about your sample transfer system. So I've seen this Leica system that you've shown. It's also used, for example, on the synchrotron when they're transferring crystals under vacuum, very similar system. So my question for you is, in the design of your system, how flexible is this? Say if you had a sample already under vacuum, would we be able to, for example, maintain it under vacuum and then put it into your system? I'm thinking more about using correlative techniques where you collect a sample already under vacuum and you want to maintain it. And is your system built in such a way that one can just use that transfer method, recover it, and put it into yours? So
yeah, absolutely. So already at MPL, I collaborate with people in the materials area where we're using this instrument to look at perovskite materials, which are very kind of water sensitive. So we do imaging in my Orbisims instrument, and then we put the sample into this vacuum transfer system, and then we take it up to a glove box they have with an AFM and other materials characterization techniques, and that glove box has got the receiving end of that docking station, and then they can then analyze the sample without it ever coming out of vacuum, and that works really well. We haven't yet done it cryo to cryo, and MPL's recently got a cryo EM, so we're just, yeah, I've been trying to get some funding to do exactly this. So to be able to do correlative cryo EM, cryo Orbisims is kind of a bit of a dream at the moment to do. Hopefully we can do that soon, but that's the idea.
we're interested in that too, so maybe I'll chat to you afterwards.
Yeah, brilliant. Yeah, that would be great.
Thank you.
Please.
I think you got quite a few new collaborators or users of the
wonderful
facility, and I think it's really cool. Kitty, are there more questions? There's some.
One
I
more.
think they all came in last but now.
Yeah. So there's one last question that I can ask is it's from Vasil Vaskiski.
Yeah, I can ask myself.
Go ahead.
Yeah, hello. So I wanted to ask about the size of the image and mass spectrometry images. So why are they limited to this small size, 128 by 128? And if it's possible to use like tile-based image capturing, like in light microscopy, to capture more information from the tissue?
Okay, yeah, no, good question. So we can drive them at any pixel density. So 128 was just what we used in that example, but 256, 512 by 512, 1024 are all possible. When you're using the Orbitrap analyzer, because it can only operate up to about 20 hertz, there is scaling things. Eventually, if you have too many pixels, it would just take you too long to take the image. So that's kind of one trade-off. Not really an issue for TOF analyzers, because you can run them so fast. But for the Orbitrap analyzers, where you've got this trade-off really between time of the analyzer and how much biological information do you want to get, 128 is, or 256 perhaps at the biggest. And then the tiling, yeah, so that's pretty standard for us. So in the TOF mode, for example, we could tile an image over a whole piece of tissue, identify that particular region that we wanted to go to, then we could drive the instrument to that region, and then zoom in, and then switch over to the Orbitrap analyzer, for instance, to go in for the real high resolution, high chemical, biological resolution image.
Thank you.
Thank you, Anne, for the wonderful talk. We are also towards the end now. We have one last question from Laura, but I guess Stephanie, it's almost 2 now. Laura, you want to ask? Go ahead.
No, it wasn't a question. It was really just to say hello and thank. I'm from the Rosalind Franklin Institute, so to say thank you to Ian for positioning our new technologies in the landscape of MSI, which is really brilliant. And if anybody has any questions for us, I've left my email, so you can get in touch directly.
I guess a lot of
thank you.
Thanks, Laura.
I guess that if people want to kind of sign in and use the system for their crazy scientific idea.
Yeah, they're our favorite kind. So if you have any crazy scientific ideas, then please get in touch.
Thank you very much.
Thanks, Ian, for a great talk, and I hope you have.
Thank
Okay, you're very welcome. Yeah, thank you for the invitation.
Well, thank you very much. It was really wonderful. Thank you.
Yeah,
this is always the awkward bit, because we can't take you for a drink or something now, or for lunch or something.
No worries.
So thank you very much. And I mean, people can kind of clap in this awkward Zoom way, but thank you very much. We really appreciate it. And I think you will hear from a few of us. So
yeah, hopefully they got my email address and just feel free to email me.
we're going to bother Kevin now.
Yes, that's it. Yeah.
Kevin Brindle. I mean, he's talked always it many times, and I've always been like, oh yeah, but we have better resolution. But I mean, now I'm looking at it, I'm thinking, ooh, this is very cool. This is fantastic. Thank you.
Thank you. Thank you very much.
Thank
Thank you.
Bye.
Bye. Bye.